my name is Ashley Howell, and I'm proposing the question, can microbiome analysis prove if the Department of Environmental Conservation, also known as the DEC's maps for shellfish harvesting, are valid? So did you know that only 1/3 of the human body is actually made up of human cells? And that can be depicted by the red section on this graph. And what are the remaining 2/3, you might ask? And that's actually microbial cells, and these make up about 100 trillion cells within the body. And microbial cells are any microorganism, fungi, or bacteria that live within the ecosystem and includes the microbiome. And a microbiome is just the total sum of all these microorganisms. And the human body has multiple microbiomes, and this includes in the gut and the breasts, and these are the most studied microbiomes in the body. However, many other species also have microbiomes, and these can both have positive and negative bacteria in them that can be harmful or beneficial to the health of the ecosystem and the individual. However, the big problem that comes into play is when humans consume a type of organism that contains a microbiome with many negative bacteria. And this is very prevalent with shellfish. So because shellfish have a very diverse microbiome, they can have many harmful and negative bacteria that exist in it. So when a human consumes a shellfish with a lot of negative bacteria, this can cause harm and cause foodborne illness within the human. So there's 3 bacteria that are known to cause harm to human health. And these include E. coli, C. diff, and Salmonella. And these are the 3 major strands of bacteria that are harmful to human health, and are known to cause nausea, sickness, and possibly death. And that has been a major issue that has been occurring in the United States. And this graph just shows the amount of deaths due to C. diff. And in the past, you can see in 1999, there's about 4 deaths per million, which is small, although it was a major effect. However, in 2010, this rose to about 20 deaths per million. And in the past, C. diff was just known to cause common foodborne illnesses, including nausea and diarrhea. However, now, in about 1999, this has been causing deaths, which is a very prevalent issue. And shellfish are known to house C. diff, and that's why I'm studying mussels. So why mussels? And the number 1 reason is because they're abundant on Long Island. So because I'm doing this project at my high school, and I have a limited time restraint, I want to not only get more information for myself, but get it for my society. So I want to study the mussel harvesting for Long Island. And in addition, mussels have a very diverse microbiome. So in a study performed by the CDC, it was found that about 487 strands of bacteria were found in the microbiome of a mussel. So within these 487 strands, it's very likely that there's about a few that are negative to human health. And this, including the C. diff or E. coli, can exist within these 487 strands. So this is one of the other reasons why I chose to study mussels. And lastly, I searched on the DEC's website for mussel harvesting on Long Island, and I found that there's a lot of graphs and charts that show the mussel harvesting areas for all over Long Island. So that's why I also chose to study, because they have a lot of information on the specific field of shellfish. So the DEC made a map for mussel distribution for all over Long Island. And this is the current guide they use for mussel harvesting throughout all of the locations. However, specifically in my study, I'll be looking at the Smithtown Bay area. And this is the actual map proposed by the DEC. And as you can see, there's 3 different sections. The red being where you can never consume these mussels, which is typically at the bottom of the graph. And then the dark blue is where there's seasonal closure, which is between May 1st and December 31st. And then the light blue is where you can harvest these mussels year-round. And the very top of the graph, you could also notice that there's the blue section, which is where there's seasonal closure. And that's notable because I'll be taking a sample from that location. And as you can see in the graph, each of the sections exist within feet from one another. So how is it possible that such different microorganisms can exist within the microbiome of these mussels so closely to one another? And also, are the DEC's maps actually correct? And because the DEC did not provide a lot of public information for us to see on how they constructed these maps, we cannot prove if these maps are actually valid, and that's what I want to do in my study. And also, I'll be using DNA barcoding in my study, which is a pretty new and invasive technology, and I'll be doing this with the help of Coulter and Harbor to help find these results. However, because it is such a new and costly technology, it's most likely that the DEC has not performed this on the mussels to get all of their results. So by doing the DNA barcoding, I will be able to reach my goal, which is to ultimately prove if the DEC's maps are correct. And first, 3 different samples were collected along the Smithtown Bay, and the first being Cargo Park. and that's the very bottom of the lake, of the bay, and that's where you can never harvest these mussels. And then the second is along the Smithtown Bay Yacht Club, and that's at the very top, and that's the section that I've noted before, where there's seasonal closure. And then the third is near Long Beach Road. And the three different sections, the first two top are placed very similar to one another to just see if the microorganisms could be different very closely to one another. And the one at the bottom is just where you can never harvest these mussels. And not only were these samples collected, but they were also sent to Cold Spring Harbor with their longitudinal lab team. So for my research topic, I chose a quasi-experimental / meta-analysis. So it was first experimental because we needed to collect these samples and send them to Cold Spring Harbor. So that's where the experimental process came into play. However, it turned into a meta-analysis when I needed to search for the harmful bacteria. So because DNA barcoding provides such a plethora of information and there's so much bacteria that could be found, like in the study about 487 were found, but there could be such a vast amount in my study. So I chose to just look at the harmful microorganisms. And then once I discovered these specific harmful microorganisms that could possibly be found, then I had to search if these harmful bacteria were located in each of the samples. So I just needed to match up the harmful bacteria to each of the samples and their location. So I used specific important databases to get all of my information. So first being the Long Island Sound study. So this was just the specific water quality of the Smithtown Bay. And it gave me background information on all the bacteria that has been found in the Smithtown Bay in the past. So because there's a lot of bacteria in the waterways and in the ecosystem, it will most likely also be found in the microbiome of these mussels. So this just provides me more background on my topic. And secondly, I used the DEC's website, and this is the more obvious site because this is where I got all of the maps from, and this is also where I searched for more information to see how they made these maps, in which I was not able to find a lot. And then lastly, I used the United States Geological Survey, which just gave me the soil and rocks of that area within the Smithtown Bay, and it also gave me the information on the specific harmful bacteria that existed within the soil and the bay to see if this matched up to the microbiome, because the environment always influences the microbiome of a specific species. And these are the raw results created through DNA barcoding. So there's three different sections, and the first being the C1s, which is Cardboard Park. So there's 4 different samples from Cardboard Park. And then the second one is D1, which is along the Smithtown Bay Yacht Club. And then the third one, D2, is along Long Beach Road. And each of these bars just show the specific samples, and the length and color represent the different bacteria in the microbiome. So the length shows the relative abundancy, and the color just distinguishes the genus, phylum, and species of each of the microorganisms. And the red are actually the bacteria that have never been sequenced at Cold Spring Harbor before, so this is just the red zone for us to look at. And this is also just the raw data formed by DNA barcoding, and as you can see, this is such a small area, but there's just a plethora of information, so this is why I only looked at the harmful bacteria, because it's virtually impossible for me to search through every single one and match them up to the colors. And this is just the overall data interpretation of all of my data. So you can see the most amount of harmful bacteria is found where you can never harvest these mussels. And there's about a few for the harvest seasonably, and there's only 2 for where you can always harvest these mussels. So we can also assume that these 2 are the least harmful to human health, and these might be the most harmful to human health. So overall, this did prove that the DEC's maps were correct. So most of the harmful bacteria were in the red zone, followed by a few in the dark blue zone, and then very, very few in the light zone, which proves that the DEC was correct in fabricating their maps. And how can this information be used for other people? So this is also very viable for environmentalists. So since this bacteria is harming human health, can it also harm the other organisms within the bay? So because this can cause foodborne illness, can it also cause illness within the species that consume these mussels that are not only us? And also this can help detect water quality. So if there's a lot of E. coli or C. diff in the water, in those microbiomes, which we found to be true, this can also be in the water quality, which could be caused by pollution or just a deposit of sewage waste in all of the data. And also this can provide a lot of information for the DEC, and this will just provide more background information on their maps, and just provide more credible information on how these maps were constructed. And they could possibly even put this on this website and publish it to the public to see how it was possible to happen. And lastly, this is also viable for scientists because it will help prevent foodborne illness from occurring on Long Island in the future, and also because all the red zones where mussels or bacteria that was never harvested before, they could possibly use this bacteria to determine new bacteria that might not have been sequenced at all, especially in Smithtown Bay by Cold Spring Harbor. So in conclusion, the DEC's maps were correct, and they can be used as a current guide to harvest mussels. However, because I just looked at the Smithtown Bay, we can also assume that all the maps produced by the DEC are also most likely correct. Although I can't verify that, we could just prove that the Smithtown Bay is just a guide for all the other maps produced by the DEC. And lastly, this just will prove its ultimate goal, which is just prevent foodborne illness from occurring by mussel harvesting.
Okay, thank you, Ashley.
How did you evaluate the sources you collected to make sure they would be credible, valid, and reliable? And which sources did you discard and why?
Well, because my project did a lot with the DEC and the CDC, I wanted most of my sources to come from those two websites. So I did a lot of information on the DEC's websites to not only get the maps, but just to look if there was background information. And then also the CDC, along with the Long Island study, which I want to make sure that they're all either government resources or scholarly articles, which the Long Island study was one, because it was published in a scientific journal. And that just gave me the background information on all the bacteria. So that's why I mostly use those type of scholarly articles and government resources. However, I also found a good website from webmd.com. However, I decided to throw it away because it was a website and it was followed by .com and not .org or .gov. So that's why I threw that one away. However, that did provide background information on the bacteria within the Smithtown Bay and its harmful effects. However, I wanted to find a more credible source.
What was the fundamental argument or idea in your research, and how does this argument or idea relate to the primary purpose of your research?
So the main argument in mind was just to prove that the DEC's maps were correct. And by doing this, this will prove its ultimate goal, which is just to prevent foodborne illness from occurring. And the argument, because the DEC did make maps for all over Long Island, I just specifically looked at the Smithtown Bay in order to prove that most of the maps from the DEC could possibly also be accurate. And also because I was able to prove that the maps were correct, this will prove that the foodborne illness will hopefully reduce if people follow these maps, which is its overall goal.
Think about the initial curiosity that led to your inquiry. What other areas of inquiry might that same curiosity lead to?
So because my study did a lot with environmental sciences and biology, this can also lead me more into the water quality area. And because I said before how it can help with environmentalists, a lot of the bacteria in the microbiome is due to the outside environment. So the water quality might be very poor in the Smithtown Bay in the areas where you can never harvest these mussels at the bottom. And I would want to look more into the pollution and sewage waste that might be into the water quality and to see if that's really affecting it. So that could be another study that I'd want to look into.
Okay, thank you, Ashley.